Hello students, we are into zoology NEET class. This is Lohita. So here we are continuing to learn about the concept principles of inheritance and variation. So in previous classes we have actually started to learn about the intergenic interactions. So which means the two non-allelic genes are interacting with each other to give one effect. Okay. So that is intergenic interactions. Two non-allelic genes are interacting with each other to show one effect. So we have already learnt about complementary genes, which is an example for intergenic interactions. Today we are going to learn few more examples related to intergenic interactions. Today we are going to learn about the supplementary genes. Supplementary genes. So supplementary genes are those genes in which one type of gene, one pair of genes, they express themselves even in the presence or absence of the other gene. Okay, they express one type of genes, one pair of genes, they express themselves in the presence or in the absence of the other gene. Okay, they express themselves in the presence or in the absence of the other gene. Okay, but the other pair, the second pair, it expresses itself only in the presence of the other gene, only in the presence of the other gene. Okay, so one type of gene is expressing itself even if the other gene is present or not, whereas the second type of gene is expressing itself only in the presence of the other gene. That is supplementary gene interaction. Okay, so let us learn this with an example. We are taking the example of coat color in mice. Okay, this is the character that we are taking into consideration, coat color in mice. So this coat color in mice is actually controlled by two types of genes, C and A. These are the two genes which control the coat color. Okay, these are the two genes controlling the coat color in mice. So students, here if there is no dominant allele for C or A, in the absence of anyhow, we have actually learnt that we should consider capital letters as dominant and small letters as recessive, right. So in the absence of capital C or capital A, both, in the absence of capital C and capital A, the mice exhibits a color called as albino, which is nothing but a white color. Okay, which is nothing but the white color. In the absence of C and A, in both the cases, the animal, it expresses albino coat color. Okay, whereas only C in absence of A, in the absence of capital A, okay, only capital C in the absence of capital A, it gives black color. Okay, it gives black color. Okay, whereas if both capital C and capital A are present, okay, if both are present, then both of them supplement each other's colors and they give an agouti color, a color which is called as agouti. Okay, then coming to the next case, if only capital A is present, okay, and C allele is absent, then also it gives albino color only, See here what are the observations that are made. In the absence of capital C and capital A, both are absent, then also albino color is expressed. In both the cases, albino color is only expressed. Okay, if only capital C is present and capital A is absent, then black color is expressed. Only C is present, A is absent, then black color is expressed. That means C is the allele which is expressing itself even in the absence of the other allele. C is present, it is expressed even if A is not present, also C is expressing the color, it is giving black color. Okay, Whereas if both capital C and capital A are present, then both of them, they will interact with each other and they both together produce agouti color. Okay. If there is only capital A, this is the second type of allele we have discussed. If there is only capital A allele and if C is not present, if C is absent, then in that case, this is the second case of the allele, if the second pair of allele expressed only itself in the presence of the other gene. Because C is absent, only A is present, it is giving albino color. Okay. So, in both the cases, see, in the absence of capital C and capital A also albino is there, and in the presence of capital A and absence of capital C also albino color is expressed. That means over here C is the allele which is expressing itself in the absence of A also, but A is allele which is expressing itself only in the presence of C allele. Okay, only in the presence of C genes A is expressed. Okay, so this is the one which is which is not expressed singly. This can express singly also. C can express itself singly also. A is not expressing itself singly. Let us make a Punnett square from this. Let us make the crosses. Let us see how is the genotype and phenotype ratio is going to be. So, students, here we are going to make a cross between the two mices. We are going to make a cross between the two mices. Okay. These are agouti and albino. We will make the crosses. Okay, between agouti and albino. Okay, we will make the cross. Agouti is going to show, it is shown in the pure case, it will be like capital C, capital C, capital A, capital A. Albino will be like small c, capital A, capital A. If we are making this cross between an agouti and albino, pure, I am sorry, capital C, small c, small a, small a. Okay, if we are making a cross between this kind of combinations, then the gametes from agouti will be capital C, capital A type of gametes only. From albino, small c, small a type of gametes only. Then the F1 generation individuals will be like capital C, capital A, small a. This individual, it will be definitely an agouti only. Okay, because capital C, capital A both are present, right? So, this individual will be an agouti. Okay. So, let us see what are the offsprings coming from the crossing between two F1 individuals. Okay. Let us see first the crossing. When F1 agouti capital C, capital A, small a are crossed with capital C, capital A, small a, which is also an F1 agouti. Okay. So, let us see the gametes coming from these individuals. Capital C, capital A, small c, capital A, capital C, small a, small c, capital A. These will be the type of gametes. 25% of each gametes will be coming. Capital C, capital A, small c, capital A, small c, capital C, small a, small c, small a. In that way, 25% of the gametes of each type, 4 different types of gametes will be produced from both the individuals. Same type of gametes will be produced. Let us now see what will be the cross after crossing the F1 and F1 two individuals, two F1 agouti individuals. Okay. So, we are now going to make this cross. Cross making is simple, but identification of the phenotype is tricky. Capital C, capital A, small c, capital A, capital C, small a, small c, small a. These are the gametes. So let us write these, what are the possible genotypes. Capital C, capital C, capital A, capital A, capital C, small c, capital A, capital A, capital C, capital C, capital A, small a, capital C, small c, capital A, small a. Let us first write all the genotypes, okay. So this with all the 4 type of gametes, capital C, small c, capital A, small c, capital A, capital C, small c, capital A, small c, capital A, small a. Now with this one, with all the 4 different gametes, capital C, capital A, small a, capital C, small c, capital A, small a, capital C, small a, capital C, small c, small a, small a. Now with this one, with all the 4 gametes, capital C, small c, capital A, small a, small c, capital A, small a, then we have capital C, small a, and we have small c, small a, small a. So these are all the different possible genotypes of the F2 individuals. Let us see what will be their phenotypes. So because here we have both capital C and capital A, both are present, so this individual will be an agouti, okay. Then here we have one capital C and one capital A, so this individual will be also an agouti. Capital C and capital A both are present, so this will be also an agouti. Capital C, capital A are present, again it will be an agouti. So capital C, capital A both are present, this will be an agouti. Small c and capital A, see small c is present, so because of capital C, dominant allele for C is not there, so A cannot express itself, so this individual is going to be albino, okay. This individual is going to be albino because there is no capital C to support its expression, so there is no color albino, white color individual is formed. Now again capital C, capital A both are present, so this will be an agouti, okay. Small c and capital A are present. Small c, capital C is not present, dominant allele for C is not there, only capital A is there, so it cannot express itself. Again this individual will be an albino, okay. Now capital C, capital A both are present, so this individual will be agouti. Capital C, capital A, this is again going to be an agouti. Capital C but small a, so there is no A for C to supplement the function, so C is expressing itself even in absence of capital A, dominant A allele, so this individual is going to be black because there is only capital C, no capital A, this individual is going to be black, okay. Again same with this case, capital C is present but there is no capital A, this individual is going to be black, okay. Now we have here in this case capital C, capital A both are present, so this individual is going to be agouti, okay. Here capital C is not there, even though capital A is present, this individual is going to be albino only, okay. Capital C, capital small a, so here there is no capital A, so capital C is only present alone, so this is going to give a black color, okay. Small c, small a, small a, this individual here all are recessive alleles only, so this individual is going to be definitely an albino, okay. So now let us find out, now we have understood the phenotypes and genotypes of the individual, now let us find out the phenotypic ratio, okay. So let us find out the phenotypic ratio of agouti is to black is to albino. Agouti is to black is to albino ratio, let us find. First let us see the number of agouti individuals. So this is 1, 2, 3, 4, 5, 6, 7, 8 and 9, right. So there are 9 agouti individuals. Let us now count the black individuals. Black, yes, 1, 2 and 3. So 3 are black individuals, right. Next the last leftover one is albino. So let us see how many are left. 1, 2, 3 and 4. So 4 are left. So in a supplementary gene interaction, the phenotypic ratio will be like 9:3:4. Okay, ratios remembering is very important, students. Remembering the ratios is important. So here in a supplementary gene interaction, in the supplementary genes, in case of supplementary genes, the phenotypic ratio will be like 9:3:4. Okay, this will be the phenotypic ratio of supplementary genes, 9:3:4. Okay, so this is about supplementary gene interaction, where one gene is expressing itself even in the absence of other gene. The second gene is expressing itself only in the presence of the first gene, and when both are present together, they are going to express a different character. Okay. The next thing that we are going to learn, students, is epistasis. Okay, epistasis is a type of non-allelic gene interaction. Okay, epistasis is a type of non-allelic gene interaction in which one gene suppresses the expression of the other gene. Okay, here it is a type of a non-allelic gene interaction, intergenic interaction, in which one gene suppresses the expression of the other gene. One gene suppresses the expression of the other gene. Okay. So here epistasis is that type of interaction in which one gene suppresses the expression of the other gene. It suppresses the expression of the other gene. One gene will suppress the expression of the other gene. Okay. So students, here the gene which is suppressing the expression, the gene which is suppressing the expression of the other gene is called as epistatic gene, is called as epistatic gene. The gene suppressing the effect of the other gene is called as epistatic gene. Okay. The gene which is suppressed, the gene which is suppressed, the gene which is suppressed, whose effect is suppressed, which is suppressed, is called as hypostatic gene, hypostatic gene. Okay. The genes, they are suppressing one gene, one type of gene is suppressing the expression of the other type of gene. Here the gene which is suppressing the expression of the other gene is called as epistatic gene, and the gene whose effect is suppressed is called as hypostatic gene. Okay. So this epistasis is also of different types, students. Let us see what are the different types of epistasis. Okay. The first type of epistasis we are learning is dominant epistasis. Okay, dominant epistasis. In case of dominant epistasis, one gene masks the epistatic gene. The epistatic gene masks the expression of the other gene when the other gene is either in a homozygous condition or in a heterozygous condition. Okay. When this particular gene is a dominant epistasis, means it masks this expression of the other gene in when the other gene is in either a dominant condition or in the recessive condition. Whatever is the condition of the other gene, whatever is the condition of the hypostatic gene, this epistatic gene will mask the expression of the hypostatic gene completely. Okay, that is called as dominant epistasis. Okay, so dominant epistasis, it masks the one gene masks the expression of the other gene, masks the expression of the other gene if it is either dominant or recessive, which may be either dominant or recessive. One gene, it masks the expression of the other gene, it may be either dominant or recessive. When this epistatic gene is consisting of its dominant allele, okay, when this epistatic gene is consisting of its dominant allele, in that case this gene masks the expression of the other gene in dominant condition or in a recessive condition. Okay, so the phenotypic ratio of dominant epistasis will be like 2L is to 1:3:1. This will be the phenotypic ratio. This will be the phenotypic ratio of dominant epistasis. Ratios are very important, students. In a dominant epistasis, the phenotypic ratio will be like 12:3:1 will be the phenotypic ratio in case of dominant epistasis. Okay. Then coming to the next type of epistasis, recessive epistasis. Okay, recessive epistasis. In case of recessive epistasis, this epistatic gene masks the expression of the other gene only when this is in a recessive condition. Okay, epistatic gene masks the expression of the other gene only when this epistatic gene is in a recessive condition. Okay. So, recessive epistasis means the epistatic gene masks the expression of the other gene, only when it is in recessive condition, okay. Only when this epistatic gene is in recessive condition, then only it will mask the expression of the other gene, okay. The other gene can be anything, it can be dominant or it can be recessive. When it is in when the epistatic gene is in recessive condition, it will mask the expression of the other gene, okay. So, the phenotypic ratio in recessive epistasis will be like phenotypic ratio in recessive epistasis will be like 9:3:4. Okay, the same as that of supplementary gene interaction, but whatever the conditions are happening here are different, but the ratio is same. Phenotypic ratio in case of recessive epistasis is 9:3:4. Okay, 9:3:4 is the phenotypic ratio in case of recessive epistasis. This is all about epistasis, students. Epistasis is here one gene will suppress the expression of the other gene. The gene which is suppressing the expression is called epistatic gene. The gene which is suppressed is called as hypostatic gene. Epistasis is of two main different types, dominant epistasis and recessive epistasis. Dominant epistasis, here one gene will mask the expression of the other gene. When it is in a dominant condition, it will mask the expression of the other gene which may be either dominant or recessive. Okay. When this epistatic gene is in dominant condition, it will mask the expression of the other gene. Whatever is the condition of the other gene may be, it may be either dominant or it may be recessive. The epistatic gene, if it is consisting of a dominant allele, then it will definitely mask the expression of the hypostatic gene. Okay. Then we have recessive epistasis. Recessive epistasis means here the epistatic gene will mask the expression of the other gene only when it is in recessive condition. Only when the epistatic gene is in recessive condition, it will mask the expression of the other gene. Whatever might be the condition of the other gene might be either dominant or it can be recessive. Okay, it can be anything, it can be dominant or it can be recessive. It will mask the expression of the hypostatic gene when this epistatic gene is in a recessive condition. Okay. And the ratio, phenotypic ratio of recessive epistasis is 9:3:4. Now we are going to learn about polygenic inheritance. Okay, polygenic inheritance. See students, in the earlier classes when we were learning about Mendel's experiments, we have learned that Mendel has given that one gene is giving the expression for purple color, one gene is giving the expression for white color, one gene is causing expression of tall, one gene is causing expression of dwarf. In that way we have learned that only one gene was causing the effect of one character. One gene was responsible for giving the effect of one character, right? But that is not true in all cases. Whatever the Mendel's expression was there regarding the expression of characters by the genes, one gene is expressing one character. It is not true in all cases. Okay, there are some instances where the expression of a particular phenotype is due to the combined effect of a few genes, okay, more than 2 types of genes. Due to combined effect of more than 2 types of genes also there are some characters controlled, okay. Due to effect of more than 2 types of genes also some characters are controlled. One character controlled by more than one type of gene, 2 or more genes, is called as polygenic inheritance, okay. So those genes, okay, those genes which control the same phenotypic character, are called as polygenes. They are called as polygenes, okay. These polygenes are actually, you know, this, if the polygenes, they are actually having an additive effect, okay. The dominant alleles in case of this polygenes, they have an additive effect, okay. They have an additive effect, they have a combining effect. The dominant alleles of this polygenes, they have an additive effect or they have a combining effect, okay. What is meant by additive effect or combining effect is nothing but the effect shown by the effect shown by the individual, the phenotypic character shown by the individual. It will be like the combination of all the different characters together, all of them together will give one effect, okay. So that is the dominant alleles will have an additive effect. So the inheritance of such kind of characters is called as polygenic inheritance. The inheritance of characters through this kind of polygenes is called polygenic inheritance. Polygenic inheritance is nothing but inheritance of the characters through polygenes is a polygenic inheritance. Like for example, see students, the example taken by Mendel was like tall is to dwarf, right? Tall and dwarf, okay. Tall and dwarf, these were the only 2 characters seen by the Mendel's plants, right? The plants were either tall or they were short, okay. The plants, they were either tall or they were very short. So there were no intermediate plants. That was in case of Mendel. Let us take the example of human beings, okay. This was the example of pea plants of Mendel. But when we take human beings for example, there is no restricted height for human beings, right. So it will be like in an average range, there will be different heights possible, right. Nobody is only short or nobody is only tall. There are different intermediate, all possible different intermediate heights are also present in human beings. So this is just because of the polygenic inheritance. Many genes together are giving the effect of tallness in our body. That is why different people are having different heights, okay. And the skin color, and you know, sometimes on these polygenes there will be effect of the environment also, okay. Here I have given you the example of human height. There are different possible heights in human beings which is caused due to polygenic inheritance, okay. And here in polygenic inheritance there is also environmental effect of environment on inheritance of the expression of the phenotype, okay. There is also the effect of environment on the expression of the phenotype, okay. For example, let us take the human skin color, okay. Human skin color in human beings. Let us just imagine that there are 3 types of genes controlling the human color. There are 3 different types of genes controlling the human color. Let us assume that they are A, B, C, okay. These are having their alleles also, right. Capital A, capital B, capital C are one type of dominant alleles. Small a, small b, small c are recessive alleles. These 3 are dominant alleles. Let us just imagine that capital A, capital B, capital C are dominant alleles. Small a, small b, small c are the recessive alleles. Small a, small b, small c, let us imagine that these are recessive alleles. Those individuals which have all the dominant alleles, capital A, capital B, capital C, those individuals having this type of all dominant alleles, he is going to be very dark. Okay, he is going to be very dark, and this very dark people are found in the very hotter regions of our world, like near the equator. Those people are very dark, like in case of Africa, right? So those people are very dark. Here there is also effect shown by the environment. Okay, so A, B, C, if all are dominant, then the person is going to be very dark. Okay, the person's skin is going to be very dark. If the condition is like A, B, C, C, so all are small, okay, all are recessive alleles, all the alleles are recessive, then the person is going to be appearing very fair. Okay, the person will have a very fair skin color. If there is an intermediate color, exactly intermediate, A, capital B, small b, capital C, small c, if this is the condition, then the individual is going to be having a intermediate color, okay, intermediate color or moderate color will be there. Okay, in this person moderate color will be there, and by using these 6 type of alleles, many different combinations of genotype can be formed, right? By using these 6 type of alleles, many different combinations of genotypes can be formed. Okay, but these different genotypes, they show different expression of colors depending upon the presence of the dominant genes. As I have told you already, dominant alleles give an additive effect. Okay, so according to the number of dominant alleles present in the individual, the skin color is going to be a dark or very light. More number of dominant alleles, more dark will be the skin. Less number of dominant alleles, less dark will be the skin. So in that way, dominant alleles in this polygenic inheritance will give an additive effect to the phenotypic character. Okay, so this is all about the concept polygenic inheritance. Here, what did we learn about polygenic inheritance? Genes which are controlling the same phenotypic character together are called as polygenes. Okay, so these polygenes, they are, if they are dominant, the dominant polygenes, alleles of these polygenes, they have an additive effect on the phenotypic character. Okay, they will have an additive effect on the phenotypic character. Let us take the example by considering tall and dwarf plants of the Mendel. Tall and dwarf plants of the Mendel are restricted heights. The plants were either completely tall or they were dwarf. There were no intermediate characters as observed by Mendel. Okay, but this is not true in all the cases, but this is not exactly happening in all the cases. So sometimes if we take human beings as an example, in human beings there are different heights possible in human beings. Okay, very many different heights are possible in human beings. In all the possible ranges, heights are present, right? So that is an example for polygenic inheritance. Okay, so which is due to the effect of cumulative effect of different polygenes. And environmental effect is also there on the expression of phenotype. Even environment is showing its effect on the expression of the phenotype. Okay. Skin color is another example for this polygenic inheritance. There are 3, let us assume that there are 3 possible types of genes for skin color. It is just an assumption, students. There are 3 possible types of genes for skin color. If capital A, capital B, capital C are dominant alleles, small a, small b, small c are recessive alleles, then in that case the person with all the dominant alleles, capital A, capital B, capital B, capital C is going to be very dark in color. Small a, small b, small c, this individual is going to be very fair in color. If the individual is having half dominant and half recessive, if it is completely heterozygote for these genes, then it is going to show a moderate or intermediate color, okay, which is exactly between the very dark and very fair colors. Different combinations of genotypes are also possible because there are 6 different types of alleles. So many other different possible genotypes are there, in which according to the dominant alleles present in them, the phenotypic effect is shown. Okay, so this is all about polygenic inheritance. Okay. Now let us learn about Plieotropy. Okay. So students, here till now we have studied that one gene, one effect we have studied. We have studied many genes, one effect. Now here we are going to study Plieotropy, where one gene is going to show many phenotypic effects. Okay, one gene controls many phenotypic effects. Okay, one gene controls many phenotypic effects. Many phenotypic effects. One gene controls many phenotypic effects. That is called as Plieotropy. Single gene, only one gene, it will be controlling many phenotypic effects. That is called as Plieotropy. Okay, generally this kind of Plieotropy, that means one gene controlling many phenotypic effects, is because of the gene controlling the metabolism. Because this gene is controlling metabolism, one gene is going to show many phenotypic effects. Okay, mostly the pleiotropic genes, mostly these pleiotropic genes, they are involved in metabolism. They are the genes which are involved mostly in metabolism. Okay, let us see the examples of Plieotropy, that is phenylketonuria, and sickle cell anemia. Phenylketonuria, sickle cell anemia. Okay, first let us learn about phenylketonuria. During phenylketonuria, actually during phenylketonuria, there is some disturbance in the production of amino acid, production of the enzyme phenylalanine hydroxylase. Okay, because of the lack of this enzyme phenylalanine hydroxylase, due to defect in some gene, phenylalanine hydroxylase gene is affected. Phenylalanine hydroxylase gene is affected. Phenylalanine hydroxylase gene is affected. When phenylalanine hydroxylase gene is affected, there is no phenylalanine hydroxylase. Phenylalanine hydroxylase is absent. Phenylalanine hydroxylase is absent. Okay, because of the lack of phenylalanine hydroxylase, which is involved in metabolism of phenylketonuria. Phenylalanine hydroxylase is involved in the metabolism of phenylketonuria. This enzyme is absent, so phenylketonuria is not getting metabolized. Phenylalanine, because this enzyme is not getting metabolized, phenylalanine enzyme is not getting metabolized because of the lack of phenylalanine hydroxylase. Phenylalanine, it is not getting metabolized. This phenylalanine, it will get accumulated in the body, and it affects the it causes mental retardation. Let us see its effect. Mental retardation, it causes the effect on skin, and hair pigmentation. It causes its effect on skin and hair pigmentation. See, this is only one gene, phenylalanine hydroxylase gene only, but when it is affected, see how many effects are caused. Phenylalanine is not getting metabolized, phenylalanine is getting accumulated, resulting in mental retardation, skin and hair pigmentation affected. Okay, so there are many phenotypic effects shown because of this one gene, phenylalanine hydroxylase. Okay, phenylalanine hydroxylase gene is affected, many effects are shown. And when coming to sickle cell anemia, okay, sickle cell anemia, okay, in case of sickle cell anemia, in the beta globin chain of the hemoglobin, in sixth position of this beta globin chain, in the sixth position of amino acid, in the sixth position of this beta globin chain, glutamic acid, it is replaced by valine. Okay, in case of the beta globin chain of hemoglobin, in the sixth position, glutamic acid is replaced by valine. When glutamic acid is replaced by valine, there is change occurring in the hemoglobin molecule, resulting in sickle cell allele, okay, HbS, this kind of hemoglobin is formed. The normal allele for hemoglobin is Hba, but this HbS allele is causing the replacement of the 6th position of the beta globin chains glutamic acid by valine. Valine is replaced by, glutamic acid is replaced by valine in the 6th position of hemoglobin. Okay. So, HbS is the allele which is responsible for it. Normal allele is Hba, but HbS allele is causing this kind of effect in the beta globin chain. So, that hemoglobin which is consisting of this kind of glutamic acid replaced with valine produced from effect of HbS allele is going to cause sickle celled red blood cells. That means the red blood cells shape is going to change, they are going to become sickle shaped. The red blood cells are going to change their shape, they are going to become sickle shaped. So, let us see what are the effects caused due to this HbS gene now. So, sickle cell red blood cells cause anemia is going to occur. They will not able to transport properly, they have low affinity for oxygen, they have very low affinity for oxygen. Okay, they result in anemia, and these sickle shaped cells, they undergo clumping and they block arteries, they will block arteries. Okay, they undergo clumping and they will block arteries. So, this single gene is causing these many effects. It is causing anemia, low affinity for oxygen, that means low efficient transmission of oxygen in the body, clumping of the sickle shaped red blood cells result in blocking of arteries. Okay. And one more thing is, if this HbS is present in a heterozygous condition, if this individual is Hba and HbS, this is the type of alleles present in the individual, because of the person being heterozygous for this allele, this sickle shaped the person who is heterozygous for this allele, he is resistant to malaria. He is resistant to malaria. Okay, he is resistant to malaria, because here malarial parasite, it affects the red blood cells, because the red blood cells of this heterozygous individual are sickle shaped. The malarial parasite, it cannot cross the membrane of this sickle shaped red blood cells. If it is crossing the sickle shaped red blood cells membrane also, it cannot survive inside the sickle shaped red blood cell. Okay. That is why this person is said to be resistant to malaria. Okay. This is called as heterozygote advantage. Heterozygote advantage. Okay. But this person is going to be absolutely normal in high oxygen affinity areas. In high oxygen affinity areas, this person is going to be absolutely normal, but in low affinity of oxygen conditions, then the red blood cells, they will become sickle shaped. Okay. That is the complexity present in this person, but otherwise, if staying in oxygen rich areas, in staying in areas with high affinity for oxygen, this person is going to lead a absolutely healthy life, and in addition he is going to be even resistant to malaria. Okay. So, this is all about polygenic inheritance. Okay. Here we have learned two examples, that is phenylketonuria and sickle cell anemia. Here there are one gene affected, many phenotypic effects are caused. Okay. So, this is all for today students. Here in today's class we have learnt about supplementary genes, epistasis. We have finished learning regarding intergenic interactions completely. With that, we have also started to learn Plieotropy and polygenic inheritance concepts also we have learnt in today's class. There is a lot more to learn in genetics. It is a very vast topic. We will learn the remaining concepts in the next class. Thank you all for joining the class. We will meet in the next class continuing the same concept. Thank you all.